last section, we want to spend a little bit of time just talking about, again, about how metabolism is regulated. And we've talked about a whole lot of different metabolic pathways, and we've talked about how they're individually regulated. But it's important to understand how these pathways interact with each other and how they're coordinately regulated by hormones, particularly by insulin and glucagon. And we've talked about a whole lot of different metabolic pathways, and we've talked about how they're individually regulated. But it's important to understand how these pathways interact with each other and how they're coordinately regulated by hormones, particularly by insulin and glucagon. So in this section, we're going to examine how this coordinate regulation occurs in the liver and the adipose tissue in 4 different situations. The first is after a meal in the postprandial situation. The second is between meals. The third is in prolonged starvation. And the fourth is in uncontrolled diabetes mellitus. So first, let's consider what happens in the fed and the fasted state, in the liver, the adipose tissue, and the muscle. What we want to characterize is the things that those tissues consume and the things that those tissues release. In the liver, in the fed state, we're going to consume glucose. Because insulin is so high, that glucose will go through glycolysis, will form acetyl-CoA. Very little of that will go through the TCA cycle to maintain energy for the hepatocyte, but most of it will be converted to triglycerides and cholesterol. In addition, it will take up amino acids and catabolize those. It will take up fatty acids as well. And those things will all be packaged. The triglycerides and cholesterol will be packaged in VLDL, and they'll be sent out for storage and consumption in the other tissues of the body. The adipose tissue will take up glucose for the sole purpose of going through glycolysis and forming glycerol in order to form triglycerides from the free fatty acids that it's going to obtain from VLDL. Muscle will take up glucose and form glycogen. It will store glycogen, as will the liver in the fed state. It'll also take up amino acids, and those amino acids will be used to add bulk to the muscle. In the fasted state, the liver is going to consume amino acids, which come from a protein breakdown and from the diet. It will consume lactate from hypoxic muscle and from red blood cells, and it will take fatty acids released from the adipose tissue, and it will take glycerol, also released from the adipose tissue. The amino acids will go through gluconeogenesis to produce glucose. The lactate, again, will be converted to pyruvate and go through gluconeogenesis to produce glucose. The fatty acids will largely be converted to ketone bodies to provide energy for the brain. The glycerol can also be converted, can also go into gluconeogenesis to form glucose. The adipose tissue releases fatty acids and glycerol under the action of hormone-sensitive lipase. It responds to glucagon. It releases the fatty acids. It breaks down the triglycerides and releases fatty acids and glycerol, which can be consumed by some tissues, but also are consumed by the liver to produce ketone bodies. The muscle tissue will produce lactate, which we've talked about already, in conjunction with gluconeogenesis in the liver. And it will produce amino acids like alanine and glutamine, which can go to the liver then and be used to form glucose in gluconeogenesis. The muscle and other tissues then in starvation can consume fatty acids, which are released from fat. They can consume ketone bodies, and they can also consume amino acids to some extent. So what happens in postprandial metabolism? So you eat a meal that's high in blood glucose, and this stimulates the secretion of insulin. So the insulin goes to the liver, it increases the glucose uptake, stimulates glycolysis, and inhibits gluconeogenesis. Remember, that occurs through that bifunctional enzyme with increasing levels of fructose 2-bisphosphate. Glycolysis then produces pyruvate and acetyl-CoA, which can go then to form cholesterol, fatty acids, and so forth. Now, the insulin also stimulates glycogen synthesis, and it inhibits glycogenolysis. Remember, by the phosphorylase kinase and the glycogen phosphorylase inhibiting glycogenolysis and dephosphorylating glycogen synthase so that glycogen synthesis occurs. Similarly, insulin leads to the dephosphorylation of acetyl-CoA carboxylase, and this leads to fatty acid synthesis. The production of malonyl-CoA by acetyl-CoA carboxylase blocks carnitine palmitoyl transferase 1 and inhibits beta oxidation. So you get a net production of fatty acids. In addition, the insulin affects the adipose tissue, as we've indicated already, by increasing glucose uptake for glycerol production and by inhibiting lipolysis. Between meals, then, after a while, the blood glucose starts to fall, and this stimulates the secretion of glucagon. Glucagon then affects the liver by stimulating gluconeogenesis. Again, that affects the bifunctional enzyme. It causes the bifunctional enzyme to be phosphorylated and decreases levels of fructose 2,6-bisphosphate. That stimulates gluconeogenesis and inhibits glycolysis. It stimulates glycogenolysis, again, by phosphorylating phosphorylase kinase and also phosphorylating glycogen phosphorylase. This leads to the breakdown of glycogen to provide glucose to maintain blood sugar. It also phosphorylates glycogen synthase, so it blocks the new formation of glycogen. Now, glucagon also phosphorylates acetyl-CoA carboxylase. This blocks fatty acid synthesis by inhibiting acetyl-CoA carboxylase. In that case, the malonyl-CoA levels fall, and when the malonyl-CoA levels are low, carnitine palmitoyl transferase is increased, and you start to get beta oxidation of free fatty acids. Glucagon also affects the adipose tissue. It stimulates hormone-sensitive lipase, which causes the breakdown of triglycerides. Now, that occurs between meals. Now, in starvation, glucagon is very high and insulin is low. So what happens, we have to understand the time course. Initially, the liver relies on glycogen stores to maintain normal blood glucose, but these stores are not very large, and they're depleted in 1 to 2 days. After that, the protein starts to be degraded, and the amino acids are transported to the liver to be substrates for gluconeogenesis. In the adipose tissue, glucagon stimulates lipolysis, and this increases the blood free fatty acid concentration. These free fatty acids are oxidized by various tissues, except for the brain. And then the liver oxidizes them to form ketones, which the brain can then absorb. So what we get in starvation is we start to see ketosis, we start to see protein breakdown in muscles and other tissues. This is basically schematized here. The fatty acids are broken down to free plasma fatty acids. That goes to plasma lipoproteins to provide the fatty acids for other tissues. It also forms ketone bodies for the brains. And then amino acids from the peripheral tissues go to the liver, and glucose is formed from those by gluconeogenesis. And that helps to maintain blood glucose over the long haul. So what are the different sources of blood sugar at any given time? Let's say that a meal was eaten at time 0, and then no meal was eaten after that for, say, 40 days, okay? Initially, in the first 4 hours or so, there's enough blood glucose from that. There's enough glucose from your meal that maintains blood glucose alone. After about 4 hours, the blood glucose starts to fall, and then we're dependent upon glycogen to start providing glucose to maintain the blood glucose. And that lasts, again, for 24 to 48 hours. After that, we have to start relying on gluconeogenesis. And remember, gluconeogenesis uses glycerol, it uses amino acids, it uses lactate, all of these things as substrates for gluconeogenesis. As time goes on, the amino acid component becomes more and more prominent. And this is just indicated again in stages in this chart. Now, what about diabetes mellitus? So in type 1 diabetes mellitus, there's high blood glucose because there's an insulin deficiency. But the lack of insulin causes cells to respond as if the blood glucose were actually very low. Now, the cells can't take up glucose due to the lack of activation of GLUT4 transporters. So we can't increase the blood glucose uptake in the peripheral tissues. So what this does is it creates a self-perpetuating spiral of hyperglycemia. So despite the fact that the blood glucose is high, the tissues respond as if there's no blood glucose. For example, in the adipocyte, there's increased blood catecholamines and glucagon, and there's a stress response that's not opposed by insulin. So this stimulates hormone-sensitive lipase. So there's lipolysis, and free fatty acids are produced. In addition to this, there's no glucose entry into the fat cells, and so there's no glycerophosphate for fat synthesis. And this aggravates the imbalance between triglyceride degradation versus triglyceride synthesis. So you have no synthesis of triglycerides, you only have degradation of triglycerides. In the liver, this high concentration of free fatty acids leads to accelerated fatty acid oxidation and very high blood ketone body concentration. In addition to that, The large influx of fatty acids results in the synthesis of VLDL, which is transported into the blood. The liver will probably have reasonable glycogen stores. The person wasn't starving, and so this will be broken down to raise the already high blood glucose levels because there's no insulin to stimulate glycogen synthesis. In addition, the other hormonal signals result in breakdown of protein, formation of glucose from the amino acids of the liver. So despite the fact that the blood glucose is actually very high, there's going to be a stimulation of glycogenolysis and a stimulation of gluconeogenesis, which produces even more glucose, causing this self-perpetuating spiral. And here's a schematic of that. So in the adipose tissue, you break down triglycerides to fatty acids. Fatty acids go to the liver where they form ketone bodies, so you get a ketosis. In addition, you get increased VLDL, increased plasma lipoproteins, and so these people are often hyperlipidemic as well. In the liver also you have stimulation of glycogenolysis, which leads to increased glucose production from the liver, and you also have a stimulation of gluconeogenesis, which further exacerbates the hyperglycemia which these patients see. So what happens in this disease is you have huge amounts of hyperglycemia. The kidneys are no longer able to reabsorb all of this, and so you lose a lot of glucose in the urine. Water follows the urine, and that leads to dehydration. The excess ketone body production causes a ketoacidosis, and the pH falls. And both of these things together lead to the symptoms of tachycardia, tachypnea, dry skin and mucous membranes, vomiting, and disorientation, confusion, and coma that result from the acidosis. You treat a ketoacidotic crisis in diabetes as follows. You give them insulin. That's the first most important thing. That's the reason they're having this problem in the first place. You give them lots of IV saline to correct the dehydration, the electrolyte imbalances, and then you have to frequently monitor the glucose and electrolyte levels so that you don't overcorrect the problems. Eventually, you may have to administer some electrolytes to correct those things. You give potassium, and you give phosphate. Now, unlike type 1 diabetes, type 2 diabetes is actually a disease of insulin resistance. There's plenty of insulin around, and in fact, insulin levels are often very elevated, but the tissues are insensitive to its action. So the result is similar to type 1 diabetes. There's this massive overproduction of blood sugar and triglycerides. However, it isn't associated with ketoacidosis. Instead, it's associated with hyperosmolar states that you get with extraordinarily high glucose levels. So with that, we conclude the regulation of metabolism section.